All right, the final part of this chapter is to talk a little bit about what happens when defects of mitosis or of meiosis occur. Actually, mainly meiosis is what we're going to cover. So, what happens when meiosis goes wrong? And so, the most common form of meiosis going wrong is called chromosome nondisjunction. So this occurs when homologous chromosomes or sister chromatids fail to separate during Anaphase I or Anaphase II. And as a result, the gametes that get produced carry an incorrect number of chromosomes. So typically they either have one too few or one too many chromosomes. And so these gametes often live, not all the time, but they often live, and so they can be used for fertilization. And so as a result, individuals that form from these gametes have an incorrect number of chromosomes, and this results in a large number of genetic disorders. So unlike other genetic disorders, this is a largely spontaneous in nature, and it's rarely inherited, and it can be recognized by karyotypes in the affected individual. So this is one of the first genetic diseases. Genetic diseases of this type were one of the first to be understood by medical doctors, and that's because they could be recognized at the level of the karyotype. So an example of nondisjunction, so this is in mitosis, and so mitosis, so we must be looking at metaphase because all of the chromosomes have aligned at the midplane. And then during anaphase, we expect each of these to split to one side. So each cell will get one copy of each sister chromatid. So what happens if something goes wrong? So if the cohesin isn't able to separate, and so instead of separating these properly, what happens is that both copies of the chromatids go to one cell, and then one cell doesn't get any of the chromatids. And so as a result, you have one cell that has one too many, and you have one cell that has one too few of the chromosomes. So if this occurs in meiosis, so in metaphase I, the homologous chromosomes, the tetrad forms, and your goal is to split the homologous chromosomes into the different cells. And so if that fails, then this will have 5, and then this will have 3. So this will have one too few, and this will have one too many. And then as we continue, this cell, so the extra homologous chromosome, that the tetrad, so what happens is the tetrad eventually is able to split apart. So even though the tetrad gets pulled to one cell, the tetrad eventually splits apart, and each one of those chromosomes is able to go to the midplane, just like it's supposed to at metaphase II. And so here you have 2 copies of chromosome 1, one here and one here, and then you have a 2, a 3, and a 4. Where in this one you have a 2, a 3, and a 4, so you're missing a chromosome 1. So once we undergo meiosis II, these copies are separated, and so again we have one too many copies of chromosome 1 here, one too many copies of chromosome 1 here, lack one of them here and here. So let's assume this occurred in the male, and each one of these is able to form a sperm. What would happen if the sperm cell fertilized a normal ovum? So if the oocyte was able to undergo meiosis II just like normal, it had its normal complement of cells. So as a result, you would have 3 copies of chromosome 1 for these, and 1 copy of chromosome 1 for these. So this would have 2 copies of chromosome 1 from the father, 1 copy from the mother, and if you added it all up, you would get 3. This would have 0 from the father, 1 from the mother, and so if you added it all up, you would get 1. So this inspires the definition trisomy and monosomy. And so a trisomy is when you have 3 of a chromosome, and a monosomy is when you have 1 of a chromosome. And so this could occur for any of the 23 chromosomes. So you could have a trisomy 3, that would just mean you have 3 copies of chromosome 3. You could have monosomy 7, and that would mean you have 1 copy of chromosome 7. If something went wrong during meiosis II, if you had a non-disjunction event in meiosis II, so meiosis I went okay, so each of these has the normal number of chromosomes, 1, 2, 3, 4, and then 1, 2, 3, 4. So what happens in this cell? So in one of these cells, one of these two daughter cells, you have a mistake during Anaphase II, and so the sister chromatids do not separate. And so this one of the cells has 2 copies of chromosome 1, and then one of the cells has 0 copies of chromosome. So in this case, you create 2 normal gametes, and then you create a gamete that has 1 too many and a gamete that has 1 too few. And so the result of fertilization would be normal, trisomy 1, and monosomy 1. So one of the first diseases, one of the first syndromes that people actually figured out the genetic basis for was Down syndrome. And so Down syndrome is a disease caused by trisomy 21. So it's associated with intellectual and physical disabilities, poor immune function, and then increased risk for other health problems. So if you have an individual with Down syndrome, so this is not the only cause, but this is the most common cause of Down syndrome. If you took a karyotype of that individual, you would, find a cell that's undergoing division, you would lyse the cell, you would stain it, and then you would line up all of the chromosomes together. So in general, most of the chromosomes would look normal. You'd have chromosome 1, 2 copies of chromosome 1. But once you got down to this chromosome 21, one of these smaller chromosomes, you would find that you actually have 3 copies of it. So the act of having your cell is not able to accommodate having 3 copies of chromosome 21, and so something goes wrong during development. So development, because of these extra copies of these genes, something goes wrong with development, so that you don't have completely normal wild type human behavior. So instead of that, you have this modified development that is then associated with intellectual, physical disabilities and other things. So in general, most of development goes right. Like, the cell is able to go to term. The cell is able to form a fetus. The fetus is able to grow. The individual is able to survive birth. The individual is able to grow. So lots of development has gone right, but there are there are aspects of development that are affected. So other diseases, so Down syndrome is one that we talked about. In general, there are other syndromes that are associated with trisomies. So Edwards syndrome, trisomy 18, fetal syndrome, trisomy 13. And so if you took those individuals with those, you would find 3 copies of chromosome 13, etc. So there's other diseases. I've never heard of them before I started teaching this genetics class. There's other diseases, other syndromes that are associated with these other trisomies. But Down syndrome is the most common of these. There's also trisomies and monosomies that have of the sex chromosome. So there's triple X syndrome, Klinefelter syndrome, Turner syndrome. So these syndromes typically influence sex-related traits because you have 3 X chromosomes or multiple X chromosomes with the Y, multiple Y chromosomes, etc. So all of these typically influence sex-related traits. But most of these you don't actually see. So there's no trisomy 1 syndrome, there's no trisomy 2 syndrome, and there's almost no monosomy syndromes, and that's because most of these actually result in miscarriage. So very often if you have 3 copies of chromosome 1 or 1 copy of chromosome 1, you actually never see individuals that have that. And that is either because, that very often is because individuals, fertilized eggs that have that particular trisomy or monosomy end up terminating, and then as a result you have a miscarriage. So what this means is that there is a significant amount of fertilizations that result in miscarriage, and this is actually something that I never really knew until I reached the age of having children, and my wife and I were trying. But many people end up having miscarriages. A lot more people end up having miscarriages than you might think. And further, the risk of both miscarriages and Down syndrome increases with the age of the mother. And so the older the mother's age is, the more likely it is that a trisomy occurs, and as a result, you either have an increase in the probability of having a child with Down syndrome, or you have an increase in the probability of having a miscarriage. And so this is just two data points. One is the rate of births with Down syndrome. And so the older the mother's age is, the more likely it is, up to 3.6% that a birth has Down syndrome. And then also the ratio of miscarriages. So in younger women, about 10% of fertilization results in miscarriages. And then as you get older and older, that rate goes up to about 40%. And so if you look at the reasons why, that's because this increase in trisomies, there's a much greater risk of trisomies being formed and miscarriages actually being caused by that. So that's something that, like, as we go to having children at later and later ages, this is obviously something that we have to worry more and more about. So you'll probably get genetic tests after you have a positive. Once you get pregnant, you'll have some sort of genetic test for the fetus, for example, to test for Down syndrome. So there's tests now available to look for trisomy 21s just using fetal DNA, for example, in the blood. And so the reason, why is there this increased rate of trisomy? So the reason for that is that it's thought to be due to the increased age of the oocyte. So how are these ovum formed? And so it's actually this development actually occurs over the course of an individual's life. So if you are 25 years old and you are wanting to have a baby, the primary oocytes that you have as a woman, those were actually produced back in your third trimester when you were actually in the womb of your mother. So your primary oocytes were actually formed way back when, before you were even born. And so these primary oocytes are in your body, and they are stuck in prophase 1. So meiosis 1 has halted at prophase 1. Once you start to ovulate, then you somewhere around 13 years old, then you actually start to release a single oocyte, and that single oocyte is able to complete development. So it's able to complete meiosis 1, so it produces a secondary oocyte and a polar body, and it produces the second oocyte is able to complete meiosis 2, and it creates a polar body and a ovum. So it's thought that the age of the primary oocyte, so if you're a 40-year-old mother, that primary oocyte was formed 40 years ago, versus if you're a 20-year-old mother, that oocyte was formed 20 years ago. So it's thought that somehow the age of the oocyte is somehow influencing how well the meiosis 1 and the meiosis 2 are able to occur. But this is actually something that's not very well understood in an area of active research. So how do you determine if your child has Down syndrome? So if you wanted to know ahead of time, before your child was born, whether or not the child had Down syndrome. So, when I had my two children, the tests, there were certain ultrasound tests, for example, that were standard. And there were certain features on the ultrasound that would say you have this probability or this probability that your child might have Down syndrome. And so if you wanted to test whether or not and know for certain, then somehow you would need to get DNA from that child, from your fetus. And so if that fetus was a trisomy 21, then you could, for example, do a karyotype. And so when my first child was born 8 years ago, the way you would test this is what was called an amniocentesis. And so an amniocentesis is using an ultrasound to identify the position of the fetus and then simultaneously injecting a needle, a very long needle, in through the uterus. So you puncture through the uterus, and the goal is to get fluid, amniotic fluid, from the womb. And so, this is obviously a very kind of rough procedure on the mother and on the child, because there is actually a certain amount of risk for the child, damage being caused by this procedure. But there will be fetal cells within the amniotic fluid that you can then test, for example, by karyotype. For my second child, certain advances were made so that you no longer even need to really do an amniocentesis. And so this discovery, this is possible because a discovery was made that there's actually DNA from the child that is in the mother's bloodstream. So if you took a blood sample from the mother's arm, you'd find red blood cells, for example, you'd find white blood cells, but you also find DNA within the blood. And so there's maternal DNA, there's pieces of DNA from the mother cells, but there's also pieces of DNA from the fetal cells. So this fetus, because of the shared blood flow between the mother and the fetus, pieces of DNA end up in the mother's bloodstream. And so what's remarkable is that the size, and it's not really understood why, but the size of the pieces of DNA is different. And so you can actually just use size criteria in order to separate out the fetal DNA from the mother's bloodstream and then test that. So this has made possible to do tests much earlier than before. For example, you can test for the sex of the baby as soon as this fetal DNA is present in the bloodstream. You can test for genetic diseases, and so this is a much less invasive type of test that you can do. And so this is cell-free fetal DNA, and it's because this is actually circulating in the mother's bloodstream. So the cffDNA originate from placental trophoblasts. So these are cells that form the placenta, but these are cells that are actually from the fetus that are used to create this placental organ. And they shed microparticles, resulting in the shedding of DNA. So the fragments are about 200 base pairs in length, which is much smaller than the maternal DNA fragment. So this is how we're able to identify them, is because we can just use size criteria. So they're present 5 to 7 weeks after gestation, so you can start to do these tests around like 7 or 8 weeks. And approximately 11% of DNA in maternal blood is fetal origin, so approximately 1 in 10 pieces of DNA. So that is the extent of Chapter 2, and next chapter we will cover the basics of Mendelian genetics.